Okay, first off, questions? Questions, anyone? Okay, starting this Friday, there will be a review starting when this class is over. This room is open on Friday after class. And I'll send out an announcement. If you can't make it, send in your questions by email or whatever. We're supposedly got everything from Google, but I want them to try to install it so I can do the Hangouts from here. I can't do it. So you can send your questions in. We've got to start thinking a little bit about Exam 3. It usually comes a little bit, Exam 3 is the hardest exam for most people because it covers the most biochemistry. Exam 4 will be during the final slot. So I think that's going to be Tuesday at 8 o'clock in the morning. Or maybe last year they changed. It had always been Tuesday at 8 o'clock in the morning. And the last exam will cover less material. And the last exam will not have a take-home portion. So it'll be just multiple choice. Okay? And so we're still a bit from Exam 3. It tends to be, well, usually you have several choices for the date, and I don't know if I can tell you when it tends to be. Another thing is I'm trying to get some students that are interested in working either a winery or a vineyard. They're going to be here through the summer. Or want to do some kind of a research project. We're working with a group that are putting in a winery, and they have a vineyard, and so we're going to be doing, basically helping them do everything. And I'd like to get a few students that are interested, maybe in viticulture or in enology. We're going down there now. I'm going down there on the weekends, and we're trying to get their wine from last year ready to bottle. And and I'll send out an email about that. So if you have an interest in that, you can email me, and I can tell you a little bit more about it. It's actually at a monastery. So they're building a new building, and in the basement they're going to have a winery. So. It's from basically the cement up in Navasota, around Navasota, somewhere between Navasota and Branham, maybe halfway. Yeah, it's really close to Washington on the Brazos. So it's about 30 minutes from here. Okay, any other questions? Yes, ma'am. No, Exam 4 is not comprehensive. Exam 4, I cannot predict what the scores will be on Exam 4 because it is during the final block, so it depends on your other final commitments. So the material on Exam 4 usually has the high, until it became the fourth exam. That's hard for me to say. It always had the highest average, okay? But I cannot make, I've never been, when the final was comprehensive, or when the final has not, the exam during the final time is not comprehensive, I cannot predict the performance. It's the only exam I cannot tell you how well the class in general is going to do on. And that's just because there's a lot of factors. You get to drop one exam. The fourth exam covers hormones, growth and development, so it is always the highest average when that was a distinct exam in class. It depends on the grade distribution. So people then will say, you know, okay, you've made maybe two Bs, two B range and a C range. So if you take that fourth exam, you can be assured that you'll get a B if you do okay on the fourth exam. But because of other commitments, finals, and other, it depends on what else you're studying for, it's all over the board. So the averages have been extremely high on the final, and they've been very low on the final. So I can't predict that. And that's even when it was a comprehensive final during the final time. It had maybe a 20-point variation. Other exams maybe have a 5-point variation. Okay? Any other questions? Okay, let me just pick up over what we discussed last time and what we're looking at. We're looking at the pressure flow hypothesis, and we're back to water potential, where the phloem loading is what we're talking about. And remember, we finished up talking about sources and sinks. So of course the source is going to be a mature leaf, a fully sized leaf that is actually producing a lot more sugar than it's using, and that sugar or carbohydrates transported as sucrose, and then this causes a water potential gradient, and then the water will move over from the xylem, and it will help to move the phloem, the sugar down in the phloem, the sucrose down in the phloem, and then it's removed at the sink, wherever that could be. And the sink could be a root, it could be a young leaf that is not making enough carbohydrate. So if you think about when a leaf starts growing, when it's very small and it grows very rapidly, it's got to synthesize a lot of cellulose. It's got to synthesize everything that it needs. So it is rapidly taking up sucrose.
Do they ever technically like switch spots? Like we mentioned in the last lecture how the potato could be the source.
Right,
The
tuber.
Potato would be a sink.
It is.
Starches. So does it flop whenever?
They can change, yes. The question is, do sources and sinks change? Yes, all the time. In the normal scheme of things, a storage organ like a tuber or a root that will be reversed when it starts growing again. Okay, so if you leave the tubers in the field, then they're going to start sprouting. So they're going to start producing shoots. So then the storage organ, the tuber, which was the sink, now becomes the source of carbohydrates for the growing sprouts. And if you've had any potatoes that, like me, and you put them in the cabinet and forget about them, come back up a week later or a month later, and they've got all those sort of long white shoots. So in that case, that tuber was the source of all the carbohydrate. So everything can switch. And then when a leaf starts becoming senescent, it may actually have some carbon moved into that senescent leaf to allow it to start mobilizing the material to move out of the leaf. Okay, so sinks and sources switch all the time. And I think we finished up with showing you how you can actually do this. We used to do this in the lab. So we have a pretty good mechanism. Because the sources and sinks can change, the phloem transport is bidirectional, which we talked about first week of class. And it can be, there's not like vascular bundles that move down or vascular bundles that move up. The phloem, the same phloem element can move things both ways, depending upon the concentration. So if we go back here, if we look at this, where we have the leaf producing excess carbohydrate, so it's the source, we could turn this upside down and make this, instead of green, make it the tuber, where then the excess carbohydrate would be. And or we could make this the tuber, say, now the leaf is gone, so it will just reverse the flow. Okay. And when we talk about hormones, we'll talk about how hormones are transported in the phloem. Remember, the phloem is living, so we can have all sorts of mechanisms to move things one way, either up or down or both ways. In the xylem, the movement basically is from the bottom to the top, and we don't have much control in the xylem because we don't have a membrane. The membrane allows us to then have proteins in the membrane, and in addition, the cell being alive means it can produce proteins, can produce enzymes, and so there's a lot more control over movement in the phloem than in the xylem.
Can you have simultaneous movement in the phloem? I always kind of wondered this, like if I'm fertilizing my plants with some plant, growth regulators, awesome, whatever, and they're also outside photosynthesizing. Are they moving sugars at the same time that either the auxin or the cytokinins are moving?
Yeah, they're moving. Yeah, so the movement of sugars has nothing to do with the movement of anything else. There's some stuff that will be moved just by the fact that it's the flow. But for hormones are mostly moved one direction because of membrane-bound proteins. So yeah, there can be things moving up. There can be hormones actually moving up as the sugar's moving down, and you'll see that that's because of membrane.
Is that within the same
In the same.
bundle?
In the same bundle, yeah. Now, just to get ahead of ourselves, if you put a membrane here, you're going to have stuff floating around in here, so it's going to have to get to that membrane to be grabbed and moved up. But when we talk about hormones, that'll become much clearer, okay? So we have this bidirectional transport. Now we wanted to finish up with the other terms that are used, which is allocation and petitioning. And allocation is what is the fate, okay? First, when this leaf is growing, it's going to import carbon so that it can grow. And then when the leaf is mature, it's going to have to carry out the housekeeping function. So the mature leaf it's going to need carbon to maintain the cell integrity, to build proteins, to build amino acids, to build proteins, to build everything that the cell needs. So the only thing then that's exported is excess carbon. So if you remember back to the chloroplast, you have that cycle turning, a very complex cycle turning for every single CO2 that's fixed. So the CO2 the CO2 fixation has to first be greater than demands for the PCR cycle, and then that carbon then is transferred out to the cytoplasm, and then that triose phosphate can be used in the cell to meet the cell's needs. And then when you've met the needs of the cell, then that excess carbon can move out. Okay, so you've got to have a good bit of sunlight for the leaf to export carbon. Okay? And so then when the leaf is really cranked up, then we're getting a lot of carbon that's transported out of the leaf. And this allocation is, or the way we discuss this is in terms of petitioning. And these are very important concepts, because if we feed radioactive CO2 to a plant, whatever the plant is, where does it end up? What percent of that CO2 ends up in the leaf? What percent of that CO2 ends up gone, respired away? What percent of that CO2 ends up in a storage organ? Which, of course, if we're trying to grow something, is what we're probably interested in. And so another term is the sink strength. So the stronger the sink, the more carbon will be allocated to that sink. So think about it. You've got a, well, I should have brought some pictures. And I've been going out every day and taking pictures of the vineyard. So now in the vineyard, we've got vines that have fairly large leaves on them, but most of the leaves are still small. So the plant is photosynthesizing a little bit, but we're still talking about most of the carbon coming up from the root system or from the stems. And I'll bring a picture next time and show you that. So what's going to control this? Hormones, of course, the turgor of the cell, environmental factors, and how close it is. Here's an example of what we can do by adding carbon dioxide. And we'll talk a little bit about supplementing carbon dioxide later. But if we look at, in this case, I think this is wheat. So we look at the grain and we look at where the allocation and petition ends up. We end up with most of the carbon in the grain and in the root, and then others, the rest of the plant. If we actually add, or at low CO2 levels, then you see you have reduction in the roots. And most of it now goes into the grain, because that's what the plant is trying to do, is to produce seed. So at the lower carbon dioxide levels, more of the carbon goes into the grain. So let's say you grow a plant in a growth chamber with reduced CO2. The plant then, from a teleological standpoint, the purpose of the plant is to produce offspring, the next generation. So the seed is going to suffer less than the other parts of the plant. Now, this is petitioning. This grain could have a lower weight. Does that make sense? So these may not be the same size seeds. But when stressed, more of the carbon is going to go to produce the seed. The last thing we want to talk about before we go on a little bit today is not a new area of research, but an area of research that we have developed a lot of tools for in the last few years. And that is xenobiotics. Because this is the way the phloem, usually the phloem, handles foreign compounds. And the interest here is developing particularly targeted pesticides of some kind or another. And so the movement of these things from one cell to another has gotten a lot of interest, because as we learn more and more about how things move, and of course we're learning all this stuff from animal biology, then maybe it's possible to make more targeted pesticides or whatever, systemic pesticides, a pesticide that actually moves, usually in the xylem, but this is more interested in movement in through the phloem, because the notion is maybe you could make something that looks similar to a molecule that's found in the plant, so an artificial hormone that will actually move, okay? And we've known about those for a long time. But actually how they move is something that people are looking a lot more at now. We have more tools. Okay, questions about that? Okay, and I threw this up. I'm going to go over this really quickly. I just want to show you something that involves what happens when we, in this case, block the xylem, and this is the disease is called Pierce's disease in grapes. And if you're familiar with Pierce's disease or not familiar with Pierce's disease, what happens is that the leaves start turning brown and become scorched. And eventually the leaves drop off. And what's interesting in here, I can't really see one, is that when the leaves drop off in Pierce's disease, the diagnostic marker of Pierce's disease is that the leaf separates at the leaf blade from the petio. So the petio still is attached to the vine. And they call those things matchsticks because it looks like a matchstick. And that's a very specific characteristic for Pierce's disease. Just to show you again, you've got the vascular bundles, and in the case of the xylem, you have these open pipes. Well, they're open in that they do not have membranes, but they do have pits at the end, or pores, and water can move. And this is from when we talked about this at the beginning of the semester. Here's what this is a xylem. This is what a xylem looks like when it's got the bacteria that causes Pierce's disease. Pierce's disease is caused by bacteria that clogs up the phloem. I mean the xylem, sorry, the xylem. And here you can see this is not a perfectly cross section. If you see there's little indentations coming out here, that's a secondary thickening. So what you're looking at are like springs inside the cell. And in this case, this cell, when a cell starts filling up, it completely fills up with the bacteria. Now, cross-section area, we're looking down now. This is xylem, and see this is the secondary wall thickening. So think about, remember the xylem has all these really large secondary thickening. So think of this as being a spring in here, which increases the surface area. And then you look in these cells, and when the cells are infected, they are filled with the bacteria. Okay? And at higher mag, what we find, the bacteria, this is the xylobacteria, it's embedded in something called a biofilm. Anybody ever heard of a biofilm?
It's like a slime, like you get it on your teeth whenever, like if you haven't brushed your teeth.
Right.
Or like you get some in the water, like water out of water.
Yeah, and biofilms are very important in disease because antibiotics do not penetrate biofilms. So if you have, it's one of the explanations about why it's so easy to keep getting sinus infections. It's because you get a bloom of the bacteria which breaks out of the biofilm. You take the antibiotic, you kill those bacteria off, but there's still bacteria that are still in the biofilm. So there's actually a couple guys on campus that are working on that. And I just want to flip down here. Here's a view of the side. So here's the side of a vessel, and you can see that it's got a lot of these bacteria in it. And let me just flip down here real quick to get the best picture. Here, then, if we look at this end, what we find is that the bacteria digest these pits. So the question we were having is, well, how does this bacteria move through the xylem? The bacteria is too big to move through it. So what we actually could see is these bacteria breaking down the pits. So basically dissolving it, and there's been a lot of work to show this is what happens. You can actually measure the enzymes. So that allows the bacteria to move throughout the xylem. And just one last one here. And so you get this, in this case, this xylem is complete, the xylem milk is completely clogged with this combination of this biofilm and bacteria. So that's one of the things that causes Pierce's disease to be such a deadly disease for vines. In fact, if Pierce's disease did not exist, the entire southeast would be producing wine grapes. And we're probably not going to see a resolution of this for quite a while. Questions about that? Just very quickly, just show you a little bit of work that we did a few years ago. You don't see much ultrastructure work. Questions before we go on to the next topic here? Maybe? Breed for resistance. There is all this work is done in California. There are grape varieties that are resistant to Pierce's disease. And there's a scientist named Walker, I think it's Walker, who has crossed this wild vitis with Vitis vinifera, which is the wine grape, classic wine grapes. And so there are now PD-resistant classic varieties. But since grapes are propagated vegetatively, you've made a cross. So now you've got half of the chromosomes, or whatever the cross was, from the Cabernet, and half of the chromosomes from or half the genes from this wild variety. And by back crossing and back crossing and back crossing, you can get something very close to a Cabernet, but you'll never get exactly the true Cabernet back out of it. And so they're at the point of making wine. These plants are grown here in Texas now, and they're making wine to see what kind of wine they make. Yes, sir?
Are there other susceptible hosts to Pierce's disease?
Other susceptible hosts?
Other susceptible that are,
Yes, there's a lot of plants that almond scorch. There's a lot of scorch disease that turns out to be due to the bacteria. And so basically, since there is a gene, there seems to be a single gene for resistance, the thing to do would be to transform grape plants with that gene. And I don't know if you guys remember Jean Gould. She was in forestry, but she has a patent on a way of transforming shoot tips with DNA. So the whole thing is you would take a Cabernet shoot tip, add the gene, and now you would get the exact same Cabernet out with one added gene. And if that gene doesn't change wine production, then you'd have a Cabernet that would be resistant. But we shouldn't be growing Cabernet in Texas anyway, so other varieties are much better. Okay, any other questions before we go on to the next topic? Okay, we're going to spend today and Friday talking about respiration and lipid metabolism. And we're going to be talking about the different pathways and how these different pathways work together, how ATP is produced, and how basically everything is made in the plant. And we're going to just spend a little bit, maybe a slide or two, on respiration rates, which will come up again when we talk about climateric fruits and ethylene. Now, this is a very, very simple overview of metabolism. Okay. We have starch we've talked about. We've talked about transporting triose We have a lot of starch that goes into cellulose production. Then we have the triose and this is glycolysis. This is glycolysis, and almost in every diagram, either glycolysis or the Krebs cycle is the center part of the diagram. And so we have we have glycolysis occurring, and then from the intermediates we have the production of, in this case, some of the amino acids. So chemic acids coming out. We have then amino acids being produced, and then we have oxen, which is a similar molecule, and other things. If we go through this pathway, we get triglycerides, we get membrane components. So the point is, these things are this is a very simple diagram of a very, very complex, thousands of reactions that are occurring. And the plant has to make everything from CO2 and water and minerals. If we go so you can see this part of the pathway. This is the very complex pathway. And so when we look at this very simple view here, you know, there's thousands and thousands and thousands of reactions that are involved. Okay. So we have made carbon, we've made carbohydrate from CO2. Now we're going to take the carbohydrate and we're going to burn it to produce energy and everything else. So it's not just to produce energy, but it's to produce everything else that the cell needs. So everyone knows basic equation. We have a glucose molecule and oxygen, and so we end up with 6 CO2s and water. So for every glucose, we end up with 6 CO2s, and that's the only thing we can do. So we do a lot of respiration measurements in plants, and we can measure CO2 production and oxygen uptake. And we have an adjunct professor who works in the USDA has a children's research center in Houston. And they actually have airtight rooms that they can put kids in for the weekend or whatever. And then there's also other places where they have these, where they can put this device on your nose and your mouth. And no matter what we do, we always produce, for every glucose, we produce 6 CO2s. We don't do anything else. Right? And plants can have all sorts of differences, and we'll talk about that a little bit later. Okay? So let's get the overview again. We've got carbon dioxide coming in. We have energy from light, and then we have the O2 coming out as a byproduct. This is photosynthesis. We have starch in the chloroplast. When there's excess carbon, it's transported out as a triose. And that triose can go through glycolysis to produce pyruvate. And then if oxygen is present, pyruvate is going to go ahead and enter the mitochondria, where the pyruvate is burned to produce NADH, FADH, ATP, and the product is CO2. If there's no oxygen here, then the pyruvate is going to be converted to ethanol and CO2. And to complete it, if we take the oxygen from the light reactions, the oxygen is needed for the mitochondria to work, for energy production. So 99.999% of oxygen that plants and animals need is for one single step in this pathway. And we're going to go over that in detail. Okay? Questions? Okay, let's start breaking starch down. We talked about starch last time as being a combination of amylo and amylopectin. And the first breakdown of these units is by an enzyme called alpha amylase. And you have a really high concentration of alpha amylase in your mouth. So as soon as you start eating things that are starchy, you start breaking them down. Most of the carbon is released as maltose, which is 2 glucose units together. Then there's beta amylase, and then there's even glucosidase, which will split it into glucose. Here's a cartoon of what the different things do. So here's a cartoon of a starch grain where you have the amylose and then remember the amylopectin chains. And so here shows you what alpha amylase does, which it cuts off these things into 2 units. So instead of 1 glucose, you've got 2 glucose units together. 2 glucose units attached is maltose. And then beta amylase then can cleave off these 1,6 bonds. So you end up then with glucose. Another way that starch is broken down is starch and phosphate can produce glucose 1-phosphate directly. Okay? So there are a lot of ways that starch can be degraded. Depends on where it is and what's degrading it. We have a great ability to break starch down. We can't break cellulose down. We have a lot of ways of breaking starch down, and we break starch down incredibly quickly. Now, we talked about triphosphate being a form of how carbon is moved in and out of the chloroplast. There is another way that this can occur, and that is that glucose can be moved through, and this is fairly new, a hexose transporter. So glucose can actually be moved and then converted into hexose phosphate. So this is the chloroplast membrane. The last time we talked about how a triphosphate goes out and a phosphate goes in to balance the phosphate needs. And now we know that there's also the ability for glucose that's made in the chloroplast to be moved out. And it's there. How active it is, it's not really known. And I think I'm going to stop there, and we'll talk about the actual pathways next time. Any questions? Okay, see you guys on Friday.